Morena Tato. My name is David Chanier from Planetary Research and Genomics Aotearoa. It's a pleasure to host you all today for a seminar. I would like to thank you all for joining. I see there's quite a few people from around the country, different institutions. I see also some familiar names from Australia, so it's good. Welcome. So the speaker today is a speaker from the National Human Genome Research Institute in Baltimore, Dr. Sergey Koren. And Sergey has been developing a lot of genome assembly tools that is widely used around the world for producing high quality genomes using long read sequencing. I can tell that even in the last couple of days, we've produced a genome with one of my students using those tools. Quite exciting. We're jumping everywhere about the stats we're getting and so on. So thanks a lot, Sergey, for joining today. Now I'll just also say hi to Anne McCartney, who's joining also today. So Anne was a postdoc in Genomics Aotearoa until last year. She worked in high quality genome project with Mihai Pop. She recently got the Rewa Genome Assembly paper published and produced. So it's very fresh. But Anne joined the NIH back in July last year, just after lockdown, and she's organized this Sergey to give this seminar today. So thank you very much, Anne, and good to have you in this call today. So here we go. So Sergey, you've got time now to give us a presentation about your genome assembly tools and how you've used that for genome assembly of human. Thank you.
Yep. Thanks everyone for joining. Sorry about the odd time. It's about 6 p.m. here, so it's a nice time for me. Hopefully 10 a.m. is not too bad of a time for you. And I'm gonna talk about the complete sequence of a human genome. So kind of the two parts to emphasize here is complete. This means no gaps, no unplaced sequences, no unknown sequences. Another important part of that sentence is of a human genome. So obviously a single genome is never going to represent the entire diversity of the human population that we have on Earth. But hopefully I'll convince you through some experiments that what we have is at least a reasonable version of a human genome and not some kind of abnormal strange result for which we could get a complete sequence just because it was so weird and looks nothing like any actual living human, making that process easier. So you may have thought that the human genome is complete. There was a big splash. Here's a picture in the middle of Bill Clinton meeting Craig Venter and Francis at the White House. And there was this human genome project, which was publicly funded, run by NIH and others, and then it competed with a private effort through Telomere Genomics, which was run by J. Craig Venter. And so the Human Genome Project selected participants and volunteers, though it's primarily based on a single male individual of African-American descent from Buffalo. About 50% of the current reference is based on his genome, while the private effort, Craig just submitted himself. It was not known at the time of this press conference that that was the case, but later it was revealed that he was the donor of the genome. So, rich guy sequenced himself essentially. But this was a big effort. Obviously, it spanned several decades, and then there was work ongoing to finish it. And so there were papers of the human genome originally in 2001, and then circa 2003, there were other papers that said, here's the complete sequence of the human genome. And despite all that effort and all the publications about it, not to denigrate this, obviously this was a heroic effort by both groups, and it was a lot of work, and they did the best that technology at the time could allow, probably much better than we would have expected was even possible. It's not actually finished. There's over 200 unresolved issues. So these are things where people have flagged either the GRC, the Genome Reference Consortium themselves, or other users of the reference have flagged essentially where sequences look strange. They either had gone back and looked at the original backs which were the layout of the, which were the original input into this human genome project effort. Or they mapped some other data sets that are looking at other humans, and it looks abnormal compared to those. And there's about 100 or more gaps in the current reference. And so this kind of gives you a visualization of the current human genome whenever there's a color change here. So here it goes black, gray, that means that there's a gap, right? And so you can see that there's no single colored chromosome anywhere. Usually they're composed of a handful, 5 to 10 contigs. This is quite a good assembly, obviously. Compared to most other mammalian genomes, this is very contiguous. That we're talking about, on the order of a few hundred contigs, which is quite good, and much better than it was in 2000, where there were thousands of contigs. And this was through essentially the efforts up to GRCH38, which is what I'm showing here, released about 6-7 years ago. There was manual effort to essentially for every gap to try to resolve every gap they could, but there's still over 100 of them unresolved. And a lot of these gaps are due to large segmental duplications that are very similar, and that's very difficult to resolve through the data. The other thing that always stands out on this plot are these white regions. So there's one on chromosome 1, another example, chromosome 9, the short arms of these acrocentric chromosomes. And so these are regions where we have no sequence at all. So really, we don't know what the human genome looks like in detail there. We just have, if you open, chromosome 13 from GRCH38, it starts off with just N and about 20 million Ns, right? So really, what's missing from the human genome? So we have an idea of what's there, right? We know, so on the short arms, these are chromosomes 13, 14, 15, 21, and 22. This is the rDNA, but the rDNA is not the only thing there. We know a little bit about the structure there as well. It's thought to be enriched for segmental duplications, but we don't actually know the sequence level information for that. And that's probably about 2% of the genome. That 45 kb, that's the size of an rDNA unit of the repeat that occurs in a long array. So it's just this 45 kb repeat over and over again. And that just to give you an idea of why it's obviously difficult to reconstruct a 10 to 45 kilobase repeat of hundreds of copies. There's the centromeric repeats. So even though they're not showing up as white here, in this constriction sites of the chromosome, they are all model based in GRCH38. So essentially, for every chromosome, you can reconstruct what the most common kind of repeats look like for that chromosome in the centromere, and you just repeat that sequence over and over and over again. But none of them are actually a true sequence from any real individual or cell line or anything like that. They're just the models, and they're just meant to avoid mismapping any short read data that came from the centromeres to somewhere else in the genome, right? And that's another about 2% or so of the genome. And this is 171 base, so much shorter repeat that organizes itself into a higher order structure that's chromosome specific, though some of them are shared between chromosomes. And then the last bit, the other white regions this is on the chromosome 1, chromosome 9, chromosome 16. And there's others that are smaller and so are less visible. They're human satellites, primarily human satellite 2 and 3. And this is a simple sequence repeat, but it occurs over very, very long stretches. So this is difficult to reconstruct because it's such a simple sequence and it occurs over 20 megabases or something like this on chromosome 1. All right, so we're missing something like 6-7% of the genome, and we don't really have a good grasp of what the sequence looks like. We kind of have an idea of what we should expect, but we don't actually know what it looks like in reality, right? And you may think, well, we know what an rDNA unit is. There is a single version of an rDNA unit. We actually submitted an assembly of one a couple of years ago based on some cell lines. So we know what the 45 kb looks like. Do we really care about the rest of them? Do we care what the 100 copies look like or how they vary? And I'm going to argue that yes, we do. There's uncaptured variation in a lot of these regions. We may be able to map here, but we just have no idea, right? We may be assembling some of these regions. These may be mappable by long reads, but without having anything to anchor to, we can't really explore the variation in the segmental duplication surrounding the rDNAs. We can't answer the question of, is the rDNA structure canonical or not? There's been some work showing that maybe it has head to tail inversions. We don't know how common that is. If you just take a random human, will they have that or not? There's a big issue with false positive calls when you're missing sequence. So if I have 10 paralogs of a gene, but I only have one of them in my assembly, then I'm just going to map every read from those 10 copies of another human to one copy. And then my variants are just going to be nonsensical. And I'm just going to have to filter them all out because they're going to have weird allele frequencies. They're going to make no sense, right? And so maybe I could have called variants had I just had the other alleles, but I can't know that until I properly reconstruct the missing genes or the missing paralogs. And then another issue that has had some demonstration in databases of contamination. If you take cells that, clinical samples where you had human genome, for example, the Human Microbiome Project, which is sampling skin. Obviously, when you sample skin, it's a pretty low microbial load community. So you get a lot of skin from the actual human who you're sampling. And so you end up with a ton of human DNA that you want to subtract. You can do this computationally where you map to the human genome and throw out anything that maps. But now you end up with things like the rDNA not being subtracted because it's not represented in the human reference or other regions that are missing. And so you end up with bacterial sequences that actually contain human DNA, human genes, which we also obviously don't want, right? And so what's the problem? In short, it's very simple. So this is just a cartoon representation of what happens with genome assembly. So we have this beautiful tree input, our nice pine, right? So what we do is we do DNA extraction and sequencing. This is this chipper in the middle here. And so it outputs little wood chips. You give all these wood chips to the genome assembly algorithms. And because all the assembly algorithms are so good, especially the ones that I worked on, you get back exactly what you started with, right? And so in short, the issue is that the repeats are very long. I gave you some of the sizes like the 45 kb rDNA, but the reads are very short, right? When we take this pine tree and we shove it through the wood chipper, it's very hard to tell what the pine tree originally looked like. And so the assembler, in a lot of cases, has no way to figure out what it looked like, and it takes a best guess. And often this best guess could be quite bad, as depicted here in this cartoon, right? But recently we've had this new era of sequencing. We have long read technologies which have really revolutionized what you can expect to get from your genome assemblies. Many of them are now rivaling what the human reference GRSH38 has produced over decades of manual finishing. And so seeing this and the advances of the long reads, we decided, hey, I bet we could finish a human genome. Let's see what we can do. Because we'd seen that we could finish bacterial genomes with long reads that were intractable with Illumina data. We'd seen that we were getting very close with smaller metazoan genomes like Drosophila. And so we thought it was time to try to select the human genome. So we essentially we decided we're going to need lots and lots of long reads, and we had to select a human genome to target. And so the T2T consortium selected this CHM13 cell line, which you can see the karyotype of here below. So the reason we picked it is because this is a result of a molar pregnancy. And so this is a result where an egg cell, which has actually no nucleus, gets impregnated by a sperm cell. And so the sperm cell divides and you end up with 2 copies of all the sperm's chromosomes and no maternal material at all. So this genome is largely homozygous, which makes the assembly problem easier. It does always have 2 copies of all the chromosomes, but the 2 copies are extremely similar, much more similar than you would see in an actual living human were you to use that. And then we picked this particular cell line because it had a low passage number and the karyotype looked pretty good and stable. And we wanted to make sure, there were a couple of these cell lines available. We picked this one because it looked like the most normal of the ones we had available. And then we decided to get essentially all the data we possibly could. We started with the PacBio CLR data. This is the kind of PacBio long reads that you're used to. They're relatively high error, somewhere 85% accuracy or so. And then more recently, Nanopore data, PacBio HiFi. These 2 were really the key, as I'll talk about later. But also we got a lot of confirmatory data to be able to validate this, such as long-range Hi-C information, long-range bio-nano optical map, strand-seq, and other data to help validate and evaluate this assembly. And so this is kind of, the kitchen sink, right? We essentially threw all the data we could at this and decided to see what we could do. But really the two key technologies that I'm going to focus on is the PacBio Hi-Fi and the Nanopore Ultra Long. And so the PacBio Hi-Fi is the most recent data that the PacBio instruments are producing. They've actually had this capability for a long time. This is how IsoSeq has always worked. But the issue was those were very short, on the order of 2 or 3 kilobases long. And so for all PacBio sequencing, you make this kind of, they call it a smart bell adapter, where you ligate the adapter on the end of your double-stranded molecule that are single-stranded, so that the polymerase can go around this circular molecule as many times as it can, essentially until the polymerase either falls off for whatever reason, or is damaged by the laser enough that it essentially dies. Right, and so the key technology changes that made this Hi-Fi possible is that PacBio can now make these circles 10 or 20 kb rather than 2 or 3 kb. So that's actually getting to a reasonable length. And they're extremely accurate because you're observing the molecule multiple times. It's the same molecule, but your sequencing is random. You can then pile all these observations on top of each other and get one very accurate version of that molecule that you saw. On the other hand, we have the Nanopore ultra long data. Rather, it's much longer than what is possible with the PacBio Hi-Fi, 100 kb plus. The record is in the megabases. It's not as accurate. It's somewhere in the mid 90s now, and it's always improving. But the power here is that these are extremely long reads. And why do we care about these two in particular? Well, there's kind of 2 ways to resolve repeats. The most intuitive, and the one that I think makes sense to pretty much everyone, is length, right? And so this is where the Nanopore reads come in. So if we have this genome here on the bottom with 2 copies of a repeat, where one copy is discriminated by these little red snips that differ between the copies. So this copy has them, the other one doesn't. If we keep sequencing Nanopore reads, they're very long, but they're very erroneous. So these little gap here represent errors in the sequencing data. And if we keep sequencing them enough, we'll get lucky, and we'll get these purple reads that are long enough that they can be anchored outside of the repeat. So even though none of the reads actually showed us what this little red bit is, right? We still know that the correct path through the genome is to go through these purple reads. We know we can't connect this purple read to this purple read because of this unique region that the read contains that looks different, right? So this is kind of simple, intuitive. Just because your read is longer than the repeat, you can resolve it. The other way to resolve repeats is with accuracy, and I think this is a little bit less intuitive. But now, we're going to look at the Hi-Fi reads in the same repeats. So now the reads are much shorter, right? So none of them are going to span this repeat. It's over 20 kb in the example, let's say. But because they're very accurate, all these reads are going to capture these little red variants, right? And so here, if we sequence the reads the same way, even though now we never had a read that could get across this repeat because it was too long, you can still know that the right path is through these purple reads to resolve this copy, and then the blue reads here to resolve this copy. And we know we can't connect the same way. We can't connect this purple read to, say, this blue read over here, because there's a variant. The red variant here is going to make it look worse when you connect this read to this read versus connecting the 2 purple reads together, right? And so as long as you can discriminate errors in your read, if these red marks were just errors in the reads versus real true differences, you can get the repeats reconstructed very accurately, even though you can't actually span them with the Hi-Fi data. And so in this way, these 2 technologies are kind of complementary to each other. So when we first started working on this project and we got our PacBio data and our Nanopore data, we kind of used an approach which took the Nanopore data first. And so we use the Nanopore data to find these kind of unique markers. So these can't be SNP level because the Nanopore is not accurate, but these are structural variants. And this is a pictorial representation of the DXZ1 array. This is a centromere on chromosome X, which was the largest repeat on chromosome X when we resolved that. And so because we see 2 reads that have this purple structural variant and another 2 reads that have these 2 yellow structural variants, we can make this kind of tiling path of the longest reads through here using these little breadcrumbs of variation. We're not sensitive enough to SNP levels, like I said, but structural variants. We can make this kind of tiling path. We know what that looks like. Obviously, this is a very noisy sequence because it's based on single nanopore reads. So then we had to spend a lot of time polishing it and making sure that the consensus sequence was actually accurate and putting reads back onto this to get called and polish. This was a big effort and was non-trivial. But nonetheless, it worked. We published closed chromosome X, a couple of years ago. It came out last year. And then in follow-on work, Lennis Logden, who is part of the T2T consortium, also finished chromosome 8 in a similar way, again, using the nanopore data kind of to figure out what the distance is between these unique structural variants or variations in the data to make a path through the complex repeat. And that came out also last year, but that's 2 chromosomes. The human genome, they took about a year each, and the human genome has 23 of them, right? So I don't want to really spend the next 20 years doing this, right? So when we got this hi-fi data, we kind of took a step back and we considered how we wanted to build and how we wanted to reconstruct these genomes. So we recently published the Canu assembler, which was targeted to essentially doing as good a job with that strategy of resolving repeats through accuracy as we could. So we tried to do everything we could to make the hi-fi data, which was already starting out as pretty accurate, even more accurate. So the couple of tricks we used is first homopolymer compression, because PacBio tends to misread homopolymers. Even if you see the same molecule 15 times, you're still not certain if that's 4 copies of an A or 5 copies of an A. It's a very common repeat type. So if you just ignore those differences, the reads will just magically look much better. There will be a lot fewer errors. There are still some, we do a little bit more read cleaning and correction. So if you see overlaps of every read, and every read in this location is saying there should be a C here, and there's only one lone read that says, no, this is a G, you can be pretty confident and say, yeah, that read is a mistake. I'm going to change that G to a C. And so it'll match everyone else. And you can then build a string graph from this, but you can build it from requiring only perfect overlaps. So in Canu and in this approach, we essentially require 8 kilobases of perfect sequence. Even if it has 1 SNP difference, we consider that not an overlap, and we don't even see it in the graph, right? And then once we started building this graph, we decided then we could look at it and use Hamiltonian walks for easy tangles. This is a little bit, this is obviously we were planning to do this in a manual approach because it's a little bit hard to encode this in the assemblers, though we're working on it. And then where we have more complex walks where it's hard to resolve the path, we were going to rely on the nanopore data to help us along. And I'll give examples of both of those as I go through here. And then because the hi-fi data is really the high accuracy data type, we're going to use only hi-fi for the consensus to generate this initial assembly. And so we don't have to worry about that long process of how do we polish the nanopore data, put other data on top of it, et cetera, because we just take the consensus of the hi-fi data, and that'll give us a very high accuracy sequence. So this was work that Sergey Nurk in our group led, and this is an example of an early version of one of his graphs. And so this is visualized in Bandage, and all the long strings are the contig that are resolved unambiguously, and then they're connected by the edges, which show how you can keep going here. And so one of the remarkable things in this graph is that it's, all the components are pretty disconnected, right? You can kind of expect that each one of these little regions here, right? So like this is probably a chromosome, this is another chromosome, but really there's not very much sequence that's similar enough by this criteria of perfect overlap we're using between chromosomes, except for this big jellyfish thing that I'll get to in a bit, right? Really even the P arm of the chromosome doesn't really look that similar to the Q arm of a chromosome, that they're connected to each other, right? Even locally, the chromosomes are the only kind of regions you get are these regions locally that are complicated. You don't really see jumps across megabases of sequence, except this one here. So if you kind of lay this graph out nicer, this is just showing you what the chromosomes look like, that indeed most of these long strings are the majority of some of these chromosomes. The little black hash marks here indicate where the hi-fi data has currently a sequencing bias. It doesn't sequence GA-rich repeats very well. And so these are cases where we had actually no hi-fi coverage, so we had a gap in our graph. So we have to manually connect those, and there's about, on the order of 20 to 30 of these throughout the genome that are indicated by these little black tick marks that you can see here. But then, there's some complexity in the chromosomes, but it's not too bad. The main complex chromosome here is chromosome 9. This is not as big as it looks. It's kind of inflated because the edges are fixed length in this visualization. So this is really only about 10 megabases of sequence, but it's a recent segmental duplication where you have to walk through this twice. So you walk in here, you have to take one path through, and then you have to pick the next walk through and then out. But it's very hard, obviously, to figure out how to phase this, right? How to connect, which path do you take on this side and this side? So this was a complex region. And then this big jellyfish was the acrocentrics. The chromosomes that contain the rDNAs. So those are really the only regions where the chromosomes are similar enough to each other to complicate this graph. So how did we simplify this graph? And a lot of this was done thanks to the work of another recent postdoc who joined our group, Miko, who developed a graph aligner, which you may have used, which is meant to align sequences to these kinds of graphs. And so for some of these examples, and so I'm now showing you real examples from this bandage graph. So the key bit to realize here is that we have coverage for these nodes. So you can see, this says 20x, 29x. So these all are at the expected single coverage. So we expect that we should visit all of these nodes once. None of them look like they're just a mistake or have one read supporting them or something like that. And if you look at this graph hard enough, you'll realize that there's only one way you can possibly walk from this yellow to this yellow, visiting every node. So if you, for example, try to go this way, then you end up stuck here and you come back out and you can't ever get back to this sequence again, right? And so there's the only correct way to walk this graph is to start, over here. So if you go, if you had walked here, like you would, like I said, would look back, you walk through this node, and out. And so these kinds of things you can resolve by the Hamiltonian walk, that visiting, as long as you have the known coverage and you're required to visit every node once, you can just figure it out by looking at it a little bit, what the path is. There's other ones that are more complicated where you can't do that. So some of them obviously look more complicated in terms of the graph structure, but they can actually be quite simple. So this is an example from chromosome X, and this actually kind of pattern, this hairpin pattern, was quite common on chromosome X. So you can see by the coverage, this node has to be traversed twice. So you would go in here, you walk through and back out. But the problem here is we don't know which way we need to walk. Do we walk in and out this way, or do we walk in this way and out? And this, this could be because of a perfect palindromic repeat, but all the cases we saw, they were actually not perfect. And by looking at the nanopore data, we could tell the difference, that the nanopore data, because it's so long compared to the sequence, so the hi-fi data is not long enough for this, but the nanopore data is, that we could see that, no, you should walk clearly in this direction. It looks much better than if you try to walk in this direction based on the nanopore alignments. And then in these kind of more complex regions, it's kind of a similar issue to what I showed in chromosome 9, just on a smaller scale. We don't know when we go from this node, we don't know if we should go this way first or we should go this way, right through this kind of fork path. And so here, because this region is short enough to be spanned by the nanopore data, if you align the nanopore data, it tells you this red path with the node numbers now annotated. So you go 1, 2, 3, 4, 5, 6, so on. And this node with a higher coverage gets visited twice, which makes sense given that its coverage is double the rest of the nodes in this graph, right? And so in this way, there are some chromosomes that we call the good, right? These have a few of these regions. They're pretty easy to resolve. You can just resolve them through the Hamiltonian path or some simple nanopore alignments. There's ones that are a bit more complicated. They have more complex structure that requires more detail. This one on chromosome 18 is right at the telomere, and that it has in the HiFi data an infinite loop because it has a large segmental duplication. You could just keep going through it as many times as you want. And so the nanopore data was key here to figure out when you stop and where you stop in this graph. And then the worst of all, I guess, is the acrocentrics. And so these are, like I said, chromosomes 13, 14, 15, 21, and 22 that house the rDNA. So kind of the interesting first thing to see in this graph that I'm going to point out is that these big messy tangles that you see 1, 2, 3, 4 of, and one of them is missing. It's disconnected from the graph because of a GA that drop out of HiFi data. But these are actually the rDNA units themselves. And so one interesting takeaway that we saw earlier on in this graph is that the rDNAs seem to be different enough that they're grouping into chromosomal groups, right? So we don't ever see overlaps between, chromosome, whatever chromosome this rDNA is, and whatever chromosome this rDNA is, which was interesting in itself, because the expectation we had going in was that the rDNAs are homogenized across the entire chromosome, across all chromosomes, though that doesn't seem to be the case. There are variations that seem to be chromosome specific in them. But then kind of the assembly question here is, these are the telomeres. We could tell that because we know the telomere sequence, right? And the question here is, so I start here, and then which way do I go through these repetitive regions, right? So do I go walk out into this one, into this one, into this one, so on, right? Any of them are equally valid from this graph, obviously. And the problem is you have to cross the rDNA. So obviously, we need really long data. This is longer than the nanopore can answer in some cases. And so we use, so these are chromosome specific cluster. And then, the recombination is actually not in the rDNA that's bringing these together, which we may have thought from the start, but really it's these segmental duplications located around the rDNAs, particularly in the distal junction, the telomere side of the rDNA that are bringing these chromosomes together. And so we use both the nanopore data, but also the Hi-C data for the long range information to confirm that, yes, if you start over here, you should end up over here based on the Hi-C, the conformation capture and interaction of these frequencies, and the interaction between this bit and this bit is much higher than, say, this bit and this bit. And so we could confirm that our reconstructions actually walked the right path through this. Right, and so essentially we had this graph, last summer, and then we spent a few months trying to make figuring out all these walks, figuring out the different connections in the rDNA, and then a lot of time validating it. And then 1 year later, we have the complete sequence of a human genome. So this just gives you a quick side-by-side between the current human reference DRCH38 and CHM13. So we have 23 chromosomes. Unfortunately, because we selected that hydidiform mole, it has no chromosome Y, it's a double X. So we don't have a completed Y right now. But for the rest of the chromosomes, we have 23 complete chromosomes. We have no unlocalized sequence, no unplaced sequence. It's about 3.05 gigabases in sequence. We have no gaps. And as best as we can estimate, the quality is extremely high, Q70. It's hard to say the quality for GRCH38 just because it's not a single human. Like I alluded to before, about half of it is from a single individual's backs, but then there's backs from other people mixed in. And so there's no original source that you could go to and say, let me sequence this and map the read and check, like, but does this actually match? Because it's a mixture of multiple people that transitions between these people along the way. Right, and so we're estimating that we're adding somewhere about 200 megabases. It depends on how you define mu, but about 200 megabases, or about 6-7% of the genome versus GRCH38. So this assembly has been released online. You can get it from our GitHub page. We released the Hi-Fi graph, so everything, all the paths we use. So you can check our work and see, did we take the right path? Do you disagree with us? And our assembly, and then the latest update is this version 1.1 that is a gapless polished assembly. Along with this, because we're trying to make this a reference sequence with resources similar to how GRCH38 has tons of resources which make it so powerful, So we include an annotation. There's a UCSC genome browser for this. We have leftover from GRCH38. And so for the rest of my talk, now that I've told you how we resolved this genome, I'm going to focus on validating it, but then also some of the new biology that we've just started to see from this having this genome. So the first part is the validation, and perhaps the easiest form of validation of an assembly is that the data the data looks like the assembly, right? So essentially you can think of it as if I were to generate sequences from my assembly, would I get the same data that I got from the real genome, right? Assuming I can capture the randomness and the errors of sequencing and quirks. And so for a large part, the answer is yes. So this top part here in pink is the coverage of the hi-fi data mapped back to the assembly. The green is the nanopore data mapped back, and you can see that the coverage is quite even and all nicely distributed, no big spikes or drops, with the exception of a couple of regions here where the hi-fi is higher and the ONT is lower, and again over here. We don't think this is an assembly issue, especially because there's this inversion, right, where one technology is over-sequencing, one is under-sequencing. These seems to be sequencing biases in the technologies in the human satellite regions. So most prominent on chromosomes 1 and 9, but also on chromosome 16. But then there are these marked with an asterisk. These are the rDNAs. And so the one caveat I'll give with this genome is that the rDNA should be thought of as a model sequence, because there's so many copies on one of these chromosomes. We know that what the repeat unit looks like there, but we just put in the number of copies we expect to see based on our estimate of coverage. We didn't actually resolve to the base level every possible copy, which is why you see some coverage drops in some of these arrays, like on chromosome chromosome 13, on 15. The ones that have shorter arrays, like chromosome 14 here, chromosome 22, the coverage looks quite good. They're pretty even. It's the ones that have the larger arrays that are still model sequences, and so you do see a little bit of coverage abnormality there. But I'd say it's pretty good. And then I said it's the sequence of a human genome, and so we wanted to convince ourselves that it's a reasonable human genome. And so I saw this quote on Twitter. Someone asked, is CHM13 a weird cell line? And the response is, it's remarkably unremarkable, which I like that statement, that it just looks like average, which is good. So this is analysis from Mitchell Volger. His preprint is linked down below there. But if you look at this large group of human genomes, the Simon's diversity panel, and you say, okay, these segmentally duplicated regions, let's say the majority of the human population has 10 copies, but obviously some humans will have 6 copies, some will have 20 copies. There's a big spread, but then what's the most common copy number you see? Say 10, right? And in majority of the cases, CHM13 represents what you would see in the majority of genomes. There's a small number where the GRCH38 and CHM13 are equal, and there's a smaller number where GRCH38 is better. This is not surprising because, like I said, it's a human genome, so obviously it's not going to have the majority allele everywhere. But it's a much better representative of the human population than GRCH38. And visualizing this another way, you can see that this is a ROC curve of how well you capture essentially the human population. That the green line up here, this is essentially picking every majority allele at every location. And CHM13 is actually quite close to that, 91 versus 96 area under the curve, compared to GRCH38, which really looks like it doesn't represent any human in the population faithfully in a bunch of these regions. And so just to give you a few examples, I'm not going to spend a lot of time on these, but when you look at the things like this here, the pink here is the CHM13 copy number, and the gray or black is GRCH38. So here you have a bimodal distribution. There's majority of the humans have this copy number. Some also have this, and GRCH has this one, which essentially is not seen anywhere else in the population. So we say clearly CHM13 is doing a better job here, and that's what's shown in these pink bars. This is a figure from Mitchell's paper, so you can look at it in detail if you want later. This case, GRCH38, CHM13 are equally good. And then in these cases, it looks like GRCH38 is a little bit more abnormal than GRCH38, CHM13 is, but here it has a higher copy number than the average, whereas GRCH38 is right around the average. And here, it's in this smaller bimodal distribution peak versus this peak for GRCH38. But it doesn't look like we're dropping copies or losing copies. In the two cases where GRCH38 is a better reference, CHM13 is abnormally strange in that it has more copies that we've represented, right? Not these kind of large systematic drops in copies that we see in GRCH38. And so we can see now, we can look at all these segmental duplications now that we have this complete genome. And so the interesting thing here is, the acrocentrics are enriched in these segmental duplications, and they share a lot between themselves, right? So essentially, there's very little novel sequence on the acrocentrics. Most of it is represented at some level in the rest of the genome, but these acrocentrics are a source of segmental duplications all over the genome, particularly on some chromosomes like 9. But it seems like there's segmental duplications from these short arms coming across everywhere in the genome, which we couldn't have seen before. And the other interesting takeaway from CHM13 is that even though it's more repetitive and it has a lot more segmental duplications than GRCH38 does, they're more diverged from each other than you would expect. So there's more perfect identity sequences in GRCH38 than for us. So that implies that the human genome may be more mappable by long reads than you would expect from GRCH38, which has more perfect repeats, probably because they were copy pasted rather than actually being resolved. And just to give you a couple more examples of the results, this is an example of what I alluded to early on in my talk, that if you're missing some of your paralogs, so in this one case, this gene, it has 23 paralogs in our assembly, less than half that in GRCH38. And so when you take GRCH38 and you map other human genomes to it, so here's the CHM13 mapped to GRCH38 and so on, you get coverage fluctuations, and you get completely nonsensical rainbow variant calls. The allele frequencies here make no sense. And essentially, you wouldn't be able to call any variants here in this gene because everything is a variant. Every base is varied, right? Now, if you take the exact same data, map it to CHM13, first, you have no variation when you map CHM13 to itself. That's good. That confirms our assembly is good. But now when you look at these, the variants are spread out. They're not, all over the place. And you see clearly there's homozygous, heterozygous allele frequencies. And this makes sense from what we see. And there may be some coverage fluctuations because some humans may have copy number variation. They may have an extra copy, another paralog versus CHM13. But you can see that there's these regions which seem to have a high rate of variation throughout. Even in the short read data, you can find that these are enriched for variation, whereas before you just had variation all over the place. So now you can call variants in sequence that you had a GRCH38, but because it was missing paralogs, you would have had to filter it or you would have had to ignore it, and now you have access to that data. If you take the 1,000 genomes data, so this is now 3,200 samples of humans, you map them all to GRCH38 and to CHM13. What you see is that you systematically have a lower error rate in the reads, so they map more accurately to CHM13 than GRCH38 across all human subpopulations, with fewer pairs mapping in incorrect orientation. So essentially, CHM13 is again showing that CHM13 better represents an average human than GRCH38. And there's about 1 million novel variants in these regions that were non-syntenic that with GRCH38. And some of these, a couple of the examples of them are shown in BNC here, do seem like they're, maybe under selective pressure. We can't say for sure. Obviously, this is very early analysis. It needs a lot more work to follow up. But potentially, we're discovering, over 1 million novel variants that may be significant that we had no access to before with GRCH38. So in summary, the human genome is finally complete. This is actually the most bases that's ever been added to a revision of a human genome throughout the entire, from GRCH38 to 37 and so on. We added these 5 new chromosome arms. There's new genes and new structures that we've uncovered. And from everything we've looked at, CHM13 is a better version of a human than GRCH38. So if you're using GRCH38, you should use CHM13 instead. And PacBio HiFi really was the key technology to build these kind of nice graphs that we could then layer other data on top, like the nanopore and the high C, to resolve the tangles and their gaps and phase some of the complex regions. So what's next for the T2T consortium, now that we finished our mission of sequencing a human genome and completing it? Obviously it's study the biology of this whole genome, but then we want to make this more routine. We're working on, we can now automatically construct about half the human chromosomes push button without having to manually look at these graphs. But then we also want to be able to do this for diploids, and pangenomes, so we can get not just a human genome, but hundreds of human genomes to represent the population. And so the Human Pangenome Reference Consortium is working on this. The T2T consortium is helping select some samples where we may want to finish some of these 250 diploid HiFi genomes and take them and make them T2T complete, finish the same way CHM13 is. But we're also looking at doing a vertebrate genome T2T with some of the common model organisms like mouse, zebrafish, and others. Because I expect they'll be just as important for research as the human genome reference has been. And so with that, I'll wrap up and I'll take questions. And I want to thank everyone who worked on this project. The T2T is a big consortium. It's an open consortium, so if you want to get involved, please get in touch. But none of this work would have been possible without contributions of all these people in the group, including Anne, who joined our group and dove right in. Essentially just pushed her in the pool and said, like, here, analyze this genome, fix this, fix that. And it was great to have her. So
thank you very much, Sergey. Fantastic talk, very clear, very exciting. Thank you again, Anne, for organizing this, and good to see you. Yeah, 2 thumbs up. And so I thank you all for attending. Thank you, Sergey, and hope to see you soon. Very good.
Yep, thanks for having me.
Yep. Bye-bye.
Bye.